now. Okay, so here we are. I mentioned a bunch of objections to natural selection. There were people that accepted it as one method, but only as one among many things that could drive evolutionary change. We mentioned Kelvin's objection that the Earth wasn't old enough for a process as Lyellian, as slow and plodding, taking tiny changes and summing them up over huge amounts of time. It didn't seem there was enough time that could produce the entire spectrum of living things from the goo to the zoo to you. You have Fleming-Jenkin arguing that favorable variants couldn't be selected for because they would have to breed with others that didn't vary in that way, and that would wash out variation in the population. You have what we were just talking about, that neither Darwin nor anybody else understood where variation comes from or how it can be passed on. And you have the fact that unlike the laws of physics, which have predictive value, you can do predictions in population genetics, and we'll talk about how you do that in the upcoming weeks. But as far as predicting the total course of life on Earth, there's just no way to do that. You could not go back 500 million years ago, watch all of the sponges and trilobites and halkierid panzer slugs crawling around on the seafloor and mathematically prove that one day the biosphere would give rise to Taylor Swift. You can do that with physics. You can predict planetary orbits, for example, hundreds of years in the future. You can't do that sort of thing with natural selection any more than you could do it with human history. This is why the astronomer John Herschel referred to Darwin's idea as the law of higgledy-piggledy. It just seemed too random. Yeah. But Kelvin's objections got knocked out beginning in 1895 when physicists discovered this new thing that they couldn't explain called radioactivity. So here you've got Pierre and Marie Curie holding up this thing that's glowing very brightly. They've discovered a few grams of a new chemical element after working their way through 10,000 tons of uranium ore, all to get just a little lump of a chemical element that they call radium. Radium is present in uranium deposits, but only in minute quantities. You might know this, it used to be used to paint glow-in-the-dark watch dials because it gives off light. There was a recent documentary, I think, called The Radium Girls, about young women that were recruited to paint radium paint onto watch dials, and nobody realized or cared about the effects of this because they ended up developing cancer. Anyway, this was new, and it turns out there are chemical elements that can decay into other chemical elements, giving off energy as they do it. And about 80% of the heat inside the Earth is due to natural radioactivity. The other 20% is left over from the Earth's formation. You know, the Earth formed basically when a whole bunch of rocks in space collapsed onto each other and formed a single body. And they fell towards each other because of gravity. And that gravitational energy had to go somewhere, and it ended up in the form of heat. You know, just as if a meteorite hits the Earth now and adds mass to the Earth, that meteorite's kinetic energy has to go somewhere and it ends up as heat. and the meteorite is glowing hot going through the atmosphere, and then forms a very hot crater on the surface of the Earth. I guess with Mjolnir in the center. Anyway, Kelvin's math was basically right, except he didn't know that the Earth had an internal heat source, that the Earth can generate heat to replenish the heat that radiates away into space. And that keeps the inside of the Earth toasty warm. It keeps the continents drifting, and it keeps the volcanoes nice and warm. So that was a factor that Kelvin had missed. Kelvin, by the way, who was very old at the time, was convinced that radioactivity was a hoax. He was also convinced that heavier than air flying machines were impossible and radio was useless. So towards the end of his life, he got to be kind of a stick in the mud. But there you have it. That's our answer to Kelvin. In fact, we can use naturally occurring radioactivity, radioactive elements in the rocks, and use that to calculate the ages of not all rocks, but certain rocks, particularly many igneous rocks, rocks that used to be molten. Darwin and company had only very imprecise guesses as to how old the Earth was. It was clear that the Earth was older than the biblical account. In fact, if you remember from the video, there was that French zookeeper named Buffon, who thought about the possibility of evolution. Buffon had actually heated an iron ball to glowing hot, measured the rate that it cooled down, used that as a model for the Earth, tried to do some math, and concluded that the Earth might be as old as 100,000 years. That's a very imprecise measurement, but Buffon, back in the 1700s, had tried to calculate and gotten an answer that the Earth was older than 6,000 years. It was a good effort for the time. Nobody had any really good way to calculate the age of the Earth, but radioactivity lets you do it, because if you have a radioactive element like uranium that decays into a non-radioactive element like lead, then inside any given rock, the ratio of lead to uranium increases with the age of a rock, and you can turn that into a calculation of the age of the rock with some fairly simple algebra. And the first guy to do this was an undergrad. Makes you wonder what you've accomplished, or me, in 1910 when he first attempted to do this, and he got the right answer. The oldest rocks that he tested turned out to be 1.6 billion years old. The oldest rocks in the solar system we've now calculated to be about 4.6 billion years old. The method has been refined a lot. Methods of analysis were still kind of crude back in 1910. But Holmes basically figured out how to do it and got the right answer, close enough to the right answer. There's some refinement since then, of course. Mendel is actually your answer to blending inheritance. Remember, I used the example of an imaginary long-necked giraffe mutant that appears and is more favored than short-necked giraffes because our long neck gets more food. But as our long neck can only breed with short-necked mates, that trait is going to get diluted out. The answer is that inheritance is not like mixing paint. It's more like mixing marbles. If you mix red and white marbles, you can still get the red marbles out. And if you've got a trait because of a gene If it's a recessive gene, it might not show up in your offspring, but the gene is still there, and it still might show up in future generations. If future generations you get a homozygote, the trait will pop back up, right? You cross a purple flowered pea plant and a white flowered pea plant, you don't get pinks, you get purples, but the white can still show up in later generations. Inheritance isn't like blending at all. And Mendel had worked that out. There's a little quote from his 1865 paper. But as I was saying earlier, he published it in an obscure journal. People didn't realize what he had done. He tried to do experiments with other plant species to confirm his results on peas, and they didn't work for chromosomal reasons that Mendel could not have known about. And then he got turned into an administrator before he really was able to follow up his results the way that ideally he would have. Okay, so what do we do between Origin of Species and 1900? Well, Lamarck's ideas came back into favor because Lamarckian evolution can happen fast. A population doesn't just wait around for a favorable variant to appear. Your short-necked giraffes are not just sitting around making do and waiting for a long-necked mutant to pop up. Giraffes can stretch their own necks and then pass on that stretch to their offspring. So giraffes can make themselves vary in favorable ways. Giraffes couldn't create their own favorable variants if Lamarck's evolution is true. And so neo-Lamarckianism was quite popular, but a fellow named August Weismann argued against it because he'd hypothesized the existence of some kind of physical stuff that carried inherited traits, and he called it germplasm. And the principle of continuity of germplasm was what he argued for. And that's the idea that whatever the hereditary stuff is in your eggs or sperm, it's set apart from the rest of the body. You can, build up big muscles all you want to, says Weismann, and there's no way those muscles can do anything to the sperm that you will pass on to your offspring if you choose, of course, to do so. Giraffes can stretch their necks all they want to while they're alive, but there's no way that their necks can influence the traits that they pass on in their eggs and sperm. Incidentally, you'll still see a term used called the Weismann barrier. It's usually used in the context of gene therapy. If you're born with a defective gene, there are still experimental ways that we can put a good copy of that gene into your body cells and cure the genetic disorder. That has been done successfully, although it's still very technically iffy. But people discuss, is it ethical to cross the Weismann barrier? We can engineer the genes in your body cells, but is it ethical to engineer genes in your ovary or testis sperm so that you could pass on a genetically engineered trait to your offspring? That would be crossing the Weismann barrier, and I think most bioethicists don't like the idea. Don't think that that is a permissible use of human gene therapy. But if you get into that issue, you'll encounter Weismann's name again, because he hypothesized this barrier between the rest of the body and the germplasm that you pass on to your offspring. And then the germplasm was discovered in 1873 by a guy named Schneider. These happen to be his drawings of, I think this was in a salamander embryo's tail. And if you remember your biology 1440, as I hope you do, you can see there's a cell in interphase on the left, and then that looks like metaphase, anaphase, and telophase. These are, I think, the earliest drawings of chromosomes. And Weismann and others concluded that whatever the chromosomes were doing, what they physically were, was the germplasm that Weismann had predicted. That whatever the chromosomes were made of, it was the stuff responsible for heredity. And hereditary substance, a material bearer of hereditary tendencies, contained in the nucleus of the germ cells, and in that part of it which forms the nuclear thread. He's talking about chromosomes there. I'm not going to say more about that, but if you really want to follow this up, you can go look up Thomas Hunt Morgan, the only Nobel laureate to ever graduate from University of Kentucky, if memory serves, who clinched the case that genes are physically located on chromosomes. And there we get into genetic linkage and genetic mapping and stuff that hopefully you've had in Biology 2490. I'll step away from that and introduce you to a Dutch botanist named Hugo de Vries. He'd read Origin of Species as a student, and he started doing experiments with crossbreeding plants, and unbeknownst to him, he ended up repeating some of Mendel's work. But he didn't work with peas. He worked with this flower. This is the evening primrose, Enothera lamarckiana. And like Mendel, he tried crossbreeding various different varieties of Enothera. And he noticed that very occasionally you would get sudden changes in color or shape from one generation to the next. He coined the word mutation to cover those. This is where the concept of mutations comes from. And de Vries argued that traits had to be inherited as units rather than as blends. So he too is arguing, based on his experiments with primroses, against Fleming Jenkins' idea of blending inheritance. In fact, these units of heredity, Mendel had just called them elements. De Vries coins the term intracellular pangenes, which eventually gets whittled down to just genes. So de Vries coins this, or the word gene, or the ancestor to the word gene, and concludes, along with Mendel, that whatever is responsible for heredity, it's little particles, it's not some kind of blending. And then Hugo de Vries and two other guys named Carl Correns and Hugo Tschermak rediscover Mendel's work in the first half of 1900, and it's still sometimes called the Great Coincidence. Because all three, but especially Hugo de Vries, read this paper and realize, hey, this is what we were looking for. Here is the statistical demonstration that traits are particulate and that they're responsible for come on, Wagner, what is responsible for them is these little elements that de Vries calls genes. And of course, the genes have different alleles, which may be dominant or recessive, and presumably you know the rest. That's the ultimate disproof of the blending inheritance objection. But at first, it was not clear how genes could be responsible for traits that vary continuously. What do I mean by that? Well, Mendel's peas, the flowers are either purple or they're white. The pods are either inflated or constricted. The peas are either green or yellow, and so on. A lot of the easy Mendelian problems, which I'm sure you did a lot of in your genetics classes, deal with traits that only have a limited number of states. Like a flower can either be purple or white, and there's no other options. Or a person can either have sickle cell anemia or not, or cystic fibrosis or not. Or a fruit fly can have eyes that are red or white. You have probably come across a lot of examples of this. The problem is that a lot of important traits that are inherited don't break down into nice, neat states. There's a strong influence of heredity on human height, for example. We will actually look at that coming up. But humans are not all either basketball centers or horse jockeys, right? We are not all either 5 feet or 7 feet. Normal human height can vary continuously on a continuous range between, I mean, even including what's considered to be medical conditions like dwarfism. You know, the height of a normal human can vary between something like 4 feet 10 inches and 7 feet 10 inches or something like that. There's a pretty wide range, and there's not just a small number of heights. Pea plants, Mendel's peas, could be either tall plants or dwarf plants, but humans are not all either tall or dwarf. There's a huge number of in-betweens. And there was a long controversy between Mendelians, who wanted to explain everything by Mendel's rules, and a group called the biometricians, who thought Mendel's work was interesting but not really relevant to the important questions. That what's more important for a lot of species are things like height, weight, body part proportions, intelligence, shades of color, all of these things that vary on continuous scales. We'll talk later as to how we deal with these continuous traits. De Vries explained mutations as sudden changes in genes in this book called Die Mutationstheorie. De Vries did not really support natural selection, though, because he thought new species could spontaneously appear by mutations in a single generation. A very interesting idea that turned out not to be quite right. But one of the things I've learned about the history of biology is that it's sometimes more important to be wrong in an interesting way than right in a boring way. In fact, I've been known to call that Wagner's Law. And De Vries would turn out to be wrong about this, but in a very interesting way. And hopefully I can get to that by the end of the course. So there's this long drawn out fight between the Mendelians and the biometricians, who by 1909 supposedly had turned the flanks and pierced the center of the army that was opposed to it. And we're skipping over a lot of complex controversies, some of which we will come back to. Suffice it to say that by about 1930, Mendelian genetics plus Darwinian natural selection produces a powerful body of theory called neo-Darwinism, or the modern synthesis. This is kind of classical evolutionary biology. It's the stuff we'll be going over for the next few weeks. It's not all there is to know about evolution. We'll look at ways in which it's been expanded. But yeah, the core of traditional evolutionary biology is what you get when you take Darwin's insights and cross them with Mendel's laws. Next week, we'll look into the nuts and bolts of how you do that. In case you have forgotten, I have had students before that seemingly did a total memory dump within about 5 minutes of taking the final exam in Biology 2490. Please do not be that guy or that gal, that person. Go dust off your old notes and take them behind the woodshed if you have to. But I'm going to assume that all of you remember the foundations of Mendelian genetics, that genes exist in one or more variant forms. They're called alleles. One allele of a gene may be dominant to the others, it may be recessive to others, or you may have situations like codominance, as in human blood types, or incomplete dominance, as in white spotting on cats. Sorry, I'm looking around because two of our cats have white spots, and I was hoping I could demonstrate on one of them. They seem to be hiding right now. And somebody was asking about change in allele frequency over time, and we'll get to that soon enough. I'm not going to answer that question yet, but we'll get to it next week. You can take members of a population, a group of individuals of the same species who are all potentially capable of interbreeding or being, related through interbreeding. You can take those individuals, count all of the alleles of a particular gene, and then calculate the allele frequency as the sum of all copies of a particular allele divided by the sum of all alleles of one gene in that one population. When a gene has two alleles, we're going to be looking at the two allele case next week. And allele frequencies are numbers between 0 and 1. And in the two allele case, the frequencies are usually indicated with the letters P and Q. If you've got more than two alleles of a gene, you can use more letters. The pros would probably just write P sub 1, P sub 2, P sub 3, ... They'd use P with subscripts. But for what we're going to do here, for now, we can start by just using P and Q. Modern synthesis evolutionary theory starts by looking at allele frequency change in a population. And I'm gonna, before I start getting into the Hardy-Weinberg equation, which we'll do on Tuesday, I want to give you a brief overview of how you can use allele frequencies by themselves as indicators of population history. Because you can sometimes study human population history by looking at human allele frequencies in human populations. One way to do this is to look at the frequencies of alleles that only appear in one copy per person. Most of your genes, you've got two copies of, one from mom and one from dad. Right. Presumably everybody knows that. But there's two groups of genes that only appear in, at most, one copy per person. The first is the genes in your mitochondria. Apparently, this is a meme now. I'm supposed to remind you that mitochondria are the power plant of the cell. Because apparently this is an example of the useless crap you learn in college. As you should know, mitochondria contain their own DNA, and that DNA is passed on uniparentally. You got your mitochondria from mom. Sperm cells do have mitochondria, but they don't end up in the fertilized egg. It may very occasionally happen, but it's not really supposed to. Which means that the DNA in all of your mitochondria you got from your mother. My son has a lot of my DNA, but he does not have my mitochondrial DNA. My mitochondrial DNA is not going any further than me. I'll get over it somehow. It'll be all right. And my mitochondrial DNA is identical with my mother's DNA, and so on. You only get mitochondrial DNA, a mother passes that on to all of her offspring. The other set is Y chromosome DNA, which a father passes on to all of his sons, right? I assume I've never checked, but I'm pretty sure I've got a Y chromosome and I got it from my dad. I could not have gotten a Y chromosome from my mother, because if my mother had a Y chromosome, she'd be my father, right? Right. My mother does not have a Y chromosome that she could possibly have passed on to me. So a father passes on Y chromosomes to all of his sons and none of his daughters, because if his daughters had a Y chromosome, they'd be sons. Okay. So alleles, DNA on mitochondria and on Y chromosomes can vary, and it can mutate like all DNA does, but it's only passed on in single copies per person. And alleles of mitochondrial genes or Y chromosome genes are known as haplotypes, right? For haplo, think haploid, right? If you're haploid, you've only got one of each chromosome. Haplotypes are alleles that only appear in single copies or single variants per person. You can't be diploid for mitochondrial genes. So haplotypes are these unique sequences that are found on genetic entities that don't recombine. This is the important thing. Y chromosomes don't normally recombine with other chromosomes. They don't do that crossing over thing. They don't break and rejoin. And so the only factor that changes a haplotype is mutation. I've got, if I have a mutation in my Y chromosome, I'll pass that on to any and all sons that I have. And maybe one of my distant descendants will have another mutation, but that's going to be relatively rare. So mitochondrial DNA and Y chromosome DNA slowly accumulates mutations, but we don't have that crossing over business to complicate matters. Haplotypes are sets of either single nucleotide polymorphisms, or SNPs, and STRs. Polymorphism just means many shapes, and a genetic polymorphism just means different sequences. I might have an A nucleotide at a certain position in my sequence, and one of you men in the class might just happen to have a G there for no particular reason other than they just go back to mutations in our ancestors. So that's an SNP. STRs, single tandem repeats, on both mitochondria and Y chromosomes, there are regions of the DNA that are just repeated, like AAT, GGC, over and over and over again. And maybe at one point in my Y chromosome, I might have 6 of a particular repeat, and one of you might have 8, and another one of you might have 12, and another one of you might have 5, and so on. These don't do anything. I don't know what my Y chromosome haplotypes are unless I send in my DNA to 23andMe and they can check it for me. And this is a map of some Y chromosome STRs. Each one of these code numbers is just the location on the Y chromosome of some area where some sequence is repeated a variable number of times. So for all I know, at position DYS447, I might have 12 repeats of some sequence, and one of you biological males might have 14 or 7 or 3, whatever it might be. So we can identify haplogroups, which just means sets of either mitochondria or Y chromosome sequences, and we give these letters. So if you look at the little colored boxes on the bottom, L1, for example, is just shorthand for a particular set of SNPs and STRs. And L2 is code for a slightly different set of SNPs and STRs. And you can see that different groups of people vary in what mitochondrial DNA haplogroups tend to be most common. Looks like in Europe, typically half the population has a mitochondrial haplogroup that's just called H. That's just short for a particular set of SNPs and STRs. Native Americans very commonly have A, B, C, and D haplogroups. That just code for 4 different sets of DNA variants in mitochondrial DNA, and so on. You can look at other patterns. These don't do anything. They don't make any real difference to how your mitochondria work, or if they do, it's very subtle. They certainly aren't markers of inferiority or anything like that. And you might notice that just about every human population is a mosaic. And there's no obvious place where you could draw lines on this map to divide people up into, fixed races or anything like that. Human genetic history is much more complicated. We're all human populations have some degree of mingling. And that's a corresponding map for Y chromosome haplotypes. Native Americans, by far the most common, is Y haplotype Q, which you also find in some Siberian populations, like one up at the very top that's labeled SL, that's the Selkup people, that are thought to be relatives of the population that migrated to North America. My dad tested it. He is bright red. That's haplogroup R1b, and you can see that's the commonest in Western Europe. No surprise there. The family tree is so white it's mayonnaise, and so on. And again, many regions of the earth have one haplogroup that is most common, but there's no obvious place on this globe to draw the lines. People have mingled so much over the past 50,000 years of our existence that there's really no good place to put the boundaries. Okay, I know I'm running low on time. I tell you what, by the power vested in me by the state of Arkansas, I'm going to pick up with haplogroups on Tuesday, because I've got something really cool to tell you about them, and I don't want to rush, because I think I've got about 2 minutes left. I will point out that this is one of the things that you get your DNA tested for if you send it off to 23andMe or another one of those companies. They can give you some insight into where your ancestors might have come from. If you, were adopted or raised in an orphanage, or if, like African Americans, you lost direct knowledge of where your ancestors came from, you can sometimes work out some of it by looking at various genetic markers, and haplogroups are one of them. I've never had mine tested, but I think I mentioned my dad has haplogroup R1b. That's just a particular set of variants on Y chromosome DNA that tends to be common in populations in Western Europe, which really was not surprising at all. Doesn't really mean much. I guess it's nice to know that we probably come from there, but I already sort of knew that. But some people do find it useful and interesting. I'm not really allowed to ask, but maybe some of you have, or your family has gotten its DNA tested and you might know your haplotypes. So we'll pick up on Tuesday with a story about haplotypes that will make your blood run cold. And I will, we'll cover that next week. By the power vested in me by the state of Arkansas, I will stop recording now.